Hello, today you get to do a lab. You get to decide what question you want to answer and set up your own experiment. And you can use either plants or animals. And as far as animals, we're talking insects. So let's take a look at this activity. Um, I went ahead and opened it up in Cami. And basically, it says you and your team. Unfortunately, you have no team, it's just yourself, but you're going to come up with an experiment to see how animals or plants respond to something that you're going to change in their environment. So hopefully you remember reading earlier in this unit about how the environment can affect our, um, our development. So right here, genes, the environment, they can affect our health depending on what we're exposed to. And so you are going to be affecting the growth of plants or insects over a 4 to 5 day period. So first you need to decide whether you want to do your experiment on plants or insects. So if you have seeds, now it says pea seeds, and what I did was I went ahead and germinated these. It took about 10 or 11 days for it to get to this point, but it says to put it in a petri dish. You really don't. You can just put it in a wet paper towel in a Ziploc bag. I don't think it's even zipped. I think it's just sort of closed but not completely sealed. And then I added some water each day to make sure that it was damp, and I had all these pea seeds germinate. So I can take it from here. You don't have to do pea seeds. If you have grass seeds or any other kind of seeds that you just found at the store, that's fine too. Um, I could take 3 of these seeds and put them in one condition, and then I could take the other 3 and put them in another condition, and that could be my experiment with these seeds. You could do more. This is from the other class, Diversity Domination, where we had to grow grass seeds and put them in different conditions. So for this one, this plant was grown with different levels of acidity, or we added vinegar. So you can see how I did this experiment. This is one idea, and it doesn't have to be vinegar. The vinegar was to simulate the acid in rain that's caused by our pollution. So, but I just wanted to show you this as an example of there's you always need what's called a control, where you can compare one experiment setup that does not have um, the variable that you're testing. So for this one, I added vinegar. So these are my experimental setups, and then this is my control. So these have vinegar and these don't. The reason I have 3 of these is because I did different concentrations of vinegar, which actually is a better experiment than just saying vinegar, no vinegar, because there's different levels of acidity in rain. This is a setup my daughter is doing just because she's growing a little garden in the back and she wanted to kind of sprout some of her seedlings. But this is a great planting option if you want to do like a setup of some seeds that you have, and you could do 9 of them as the control and 9 of them as your experimental if you're adding some sort of um, exposure to something. Or you could do 3 groups. One could be your control, one could be one concentration of your variable, and then the next one could be your slightly higher concentration. So I'm just showing you these as examples. It just really depends on, what you have at home and how elaborate you want to make your setup. Um, Now, some of you might be wanting to do insects, and that takes a little collecting, so I'm not sure. I guess you can get crickets and mealworms at the pet store, but I personally was like, well, what kind of experiments can you do with insects? So I looked this up, and I'll actually put this link in your Power School. But here's some kind of bug ideas. Um, it doesn't have to be pill bugs, but this experiment is trying to see, you select different things to change in their environment. So if you want to learn more, you can click on this. This one stood out to me because a lot of times we try and repel ants by spraying them, but then those sprays can be harmful. So this is kind of looking at what are some natural repellents that might keep ants away. So if you click on this, there's a whole list of different things that you can try that are not toxic. Maybe they're not toxic to pets or young children. So here's another example you could use where you have ants, and you'll have to expose some to whatever repellent and see how they respond. So this will be interesting. I'm not sure about how easy it's going to be to get fruit flies, but it's just to kind of give you some ideas of what sort of things you can test. So this one is testing how temperature affects fruit fly development. So if you have different insects and you have the ability to change the temperature of some of them and others of them, and maybe some of them are, you know, eggs or something like that, then you could try something like that. So it's really up to you. I think the easiest ones might be, insects are a little hard to wrangle. I've done worm experiments where I test like what type of environment they like, different levels of moisture, different levels of darkness. But I would like to go over real quick what makes a good experiment, because you're going to have to fill some of this stuff in your lab. So when you do an experiment, you always want to have at least two groups, and one of them is going to be called a control group. So if I were doing an experiment to see how do plants respond to Miracle-Gro, I would have one plant or more than one plant without Miracle-Gro so that I could compare the plant that got the Miracle-Gro. So this is called my control, and then this is called the experimental group. And I think in your lab write-up, you need to know what a control is. So that would be, the plant that doesn't have the vinegar, or the ants that don't have the repellent. And so what you need to do is you always just need to have one variable that's different. Everything else needs to be the same. So for this experiment, it's the Miracle-Gro. That is the one variable. It's not that you're doing Miracle-Gro plus more water, plus more sun, plus, a different kind of seed. Everything needs to be the same, and the only difference is going to be the Miracle-Gro. So we call that the independent variable, the one thing that you're changing. And you can give different concentrations of that variable. So you could do like a small amount of Miracle-Gro, a medium amount, and a large amount. And of course, you have to measure it out, not just call it small, medium, and large. But that is still your independent variable. And then everything else is going to be the same. Same amount of water they get, same amount of sunlight, same amount of soil, same type of plant, same number of seeds that you plant. So those are all constants. And in your lab, I think, so you're going to say, what is the one thing you're going to control? What's the one thing you're going to test? So that's your variable. So is it going to be light? Is it going to be water? Is it going to be a food source, a pollutant? And by pollutant, it could be vinegar, it could be salt, it could be oil. Those all go into the environment. it could be trash, it could be detergent. So those are all different pollutants that you could test. And then you're going to write your hypothesis. What do you think? What do you think is going to happen? How do you think that this variable you test is going to affect your plants or your bugs? Okay, so those are some terms that you need to know. Hypothesis right here, plants will grow taller when given Miracle-Gro. So that's my hypothesis. And then you're going to collect data. So depending on whether you're doing plants or insects, you need to find a way to collect your data with the dates, the number of different setups that you have, and preferably have some data that involves numbers. You don't want to just say, looks good, growing well. You want to tell me, for instance, if this were your experiment, you want to tell me how many sprouted, how many germinated, how tall are they, things like that. And then you want to find a way to graph your data. And then you're going to analyze it. Okay, so for your experiment, you need to figure out what you're going to do, what your purpose is, what you want to find out, and then what the variable is that you're going to be testing, what your control is, it's going to be the one without that variable, and what's your hypothesis. Then you're going to tell me how you're going to make observations and collect data. You're going to tell me a step-by-step, what you're going to do. So you could tell me, for example, if you're doing plants, I'm going to make sure they have enough water each day, give them the same amount of water, and then every 3 days I'm going to give this one 5 milliliters of vinegar, this one 10 milliliters of vinegar, and this one 15. So that somebody can repeat your experiment by just looking at your instructions. Okay, and then any supplies, create a data table either here or on the other space. And what you're going to do is you're going to submit this to me, and I'm going to say, go for it, yes, looks good, or I'm going to give you some suggestions on how you can make it a little clearer or easier for yourself. Then you're going to, for the next week or two, you're going to be recording your data, and then you're going to write your response, what happened. Okay, what changes would you make? What were the results? Were you correct in your hypothesis? And then here you're going to go ahead and answer these questions below. Okay, so if you have any questions, please reach out to me. Otherwise, go ahead and decide, fill in these first 2 pages, submit that, and I hope you have fun with this. Okay.